Io lascio.
Addio, ciao.
Okay.
Bye.
Good afternoon, everybody. Welcome to the seminar. My name is Angela Sabino, and it's my great pleasure to introduce the speaker of the seminar today, Dr. Yolanda Sarno, who is a great collaborator and a great friend of mine. We met back in the days, like around 10 years ago, when she was doing her master thesis in Milano Bicocca. She did a master thesis in medical biotechnology, and then we met and became science buddies that we are still today. We share many scientific interests in common, and also the lab space. So we actually share our lab life together. So I will tell you about. I would like to tell you many things about her personality, but I will stick to science and her scientific journey. To make it short. So she graduated in medical biotechnology in Milano Bicocca, and then she enrolled in, when did you graduate? 2017? No, 2016. And then she enrolled in the DMET program.
DMET 2012.
DMET 2012, well, maybe the dates are not correct. But anyway, she enrolled in the DMET, Molecular and Translational Program, as a PhD student. And during this PhD experience, she had the chance to spend 6 months in the United States. And in the lab of the Dr. Cara Davis, who was a young lab back in the days and was stemming out of a bigger laboratory held by Professor Gary Nolan, who was actually the inventor of the cytometry technology that she will explain in detail in the seminar. And this technology was central in the studies of Yolanda, in the research that Yolanda is conducting. So this experience was very precious for her during the PhD, because it was not that common back in the days to go abroad during the PhD. Now it's more common for the PhD student. Now you can have this chance more easily. So after the PhD, she came back to this lab for a long postdoc. This postdoc was around 7 years. And it was a very fruitful time. And then also she tried using cytometry during this postdoc to dissect the complexity of B-cell acute lymphoblastic leukemia in children. And this postdoc was very fruitful. She did many scientific accomplishments, including scientific publication in prestigious journal, as in Nature Medicine, Nature Communication, and then she will tell you in detail later. After that, she came back. She relocated to Italy in 2023 as awardee of the ARC Startup Grant. And that is a grant that allows young researchers to build up their own research group. And in 2024, she became a researcher for Università of Milano Bicocca, so as in RTDA. I don't know how to say it in English, but it's a researcher, and she has her own research group as a young group leader in Università. So she will tell you about your research in detail. The stage is yours.
Thanks. Thank you, Angie, for the introduction, and of course the organizer for inviting me to tell you today a little bit my scientific journey from my PhD to nowadays. As Angie has said, during my scientific journey, I mainly have worked with mass cytometry, which is a relatively new technology. So today I decided to organize the seminar, starting exactly from explaining what is this technology, and then I will bring you some example of how I apply this technology to answer biological questions related with leukemia. And then finally, I will just give you a snapshot of what we are working on now in the lab as a very preliminary, let's say, data. So the cytometry is basically a mass spectrometer. It's where we have a plasma generated by an electromagnetic field. So it's an ICP source, and is a TOF type of mass spectrometer. It works in a specific atomic range between 79 and 209, giving in this way theoretically 135 channels that are open for the detection of our metals of interest. How we introduce the cells is a single cell suspension, and with the last version of the cytometry that you are seeing here, actually it's possible to do batch acquisition. So we have actually a carousel where we can load up to 13 samples, and this allows you to run multiple samples at the same time. The company claims 24 hours per day, 24 hours per day. It's way more complicated than that, but let's say that you can do very long run multiple samples. The output is an FCS file, so basically a format that is common for every flow cytometer, so you can basically analyze the data that you obtain either with any flow cytometer software. I just wanted to put you in a slide to tell you it's a new technology, but not that new. So it was born in 2009, so now it's been around for about 15 years. And as Angie has said, I had the chance to be in one of the labs that was pioneering this technology, and actually had the opportunity, great privilege of working with all of these different instruments and learning from the very beginning to the more sophisticated one that we have nowadays. An interesting step in the generation of this is that in 2015, with the Helios mass cytometer, which was the third generation, let's say, they introduced also an imaging system that allow you to do all the analysis that we do in single cell suspension, also on a slide that can be a slide of your tumor, or let's say whatever your tissue of interest is. So they will give you also the image with all the information of the antibodies. The one that we obtain in the lab, that we have nowadays in the lab, is the Cyto-XT that we purchased about a year ago, that is now up running, and I will tell you a little bit more how it works. So the main difference between mass cytometry and flow cytometry is basically the method of detection. So in both technologies, we have an antibody that is conjugated with basically the detector that we will analyze with our technology. While in flow cytometry, the antibody is conjugated to a fluorophore that is excited by the laser of the flow cytometer, and then we measure the spectra emission of this fluorophore. In the cytop, we have antibodies conjugated to metal isotopes that belongs to the group of the lanthanides. What we measure is the atomic mass of these basically metal isotopes. The main difference is that it's not a technology based on fluorescence, so therefore we don't have all the problems related with that. So we don't have the problem of autofluorescence of the cells, and also when we start to increase the number of markers that we analyzed, we don't have the problem of worrying about spectral overlap. Here the spectra are very discrete, and these allow you basically also to do very nice studies of low abundant protein. How it works, the system, I don't have the pointer, but I'll try. So basically we introduce the cells. Do you see my pointer? No. Here. So you introduce the cells using basically your suspension cells using a nebulizer. So the cells are then unified they pass through a capillary and then they form a cell aerosol. At this point, they enter in a system where there is a spray chamber that gets very, very high temperature. And then the plasma ICP source that we have, which is this plasma generated by a electromagnetic field, the cells are completely basically destroyed inside the system. And the plasma goes to ionize all the different ions that are present of the metals conjugated with the antibodies that we use in our staining. And all these atomic mass are filtered based on their mass and charge, and they are quantified passing through a TOF mass spectrometer. So the ions, the lighter ions will fly faster than the heavier ions. So in this way, we can analyze for each event passing through the machine all the different atomic mass that are related with the metal that is associated with the antibody that we use in the panel. So what we get at the end, actually the signal, when you run the samples, you see just a bunch of dark dots, let's say, but then likely, so everything digitalized. So what we get is the FCS file, and then basically you can analyze the data as you will do with any other flow cytometer. The protocol, I wanted to just give you how you prepare the samples for this. It's basically very, very similar to what you will do for a classic phosphoflow experiment by flow. So you can start from live cells, frozen cells. You can stimulate your cells with drug or cytokines. So we then proceed with the steps of with the PFA to cross-link the protein. And then we proceed with the staining, which is a two-step staining. We first use antibodies that are directly against antigen that are expressed on the surface of the cells. Then we proceed with the cell membrane permeabilization using methanol, and then we perform the intracellular staining using antibodies, this time directly against antigen that is expressed inside the cell. And then, as I told you, this cell suspension is then introduced in the system, and then we get to the maybe most difficult part of all this, which is the analysis of the data. So the panel design, so before starting the experiment, you have to spend a pretty good amount of time setting up the panel, because this is a targeted technology, so it's not that you run like RNA-seq, you put your sample, so you see what you get. In this case, you have to choose what you want to look, basically. So it requires a little bit of troubleshooting in the beginning, knowledge of what you want to analyze. Nowadays, we have 58 open channels that you can use basically to put your marker of interest, and 6 of them are dedicated to barcoding. So basically, you have the possibility of using a combination of 6 methods to barcode your samples. In this way, you can do all the process that I showed you before of staining in one tube of 20 conditions altogether. And also the run in the machine is done all together. And then after the run, you debarcode and you will get back your 20 condition. In my experience, I've used a lot of this opportunity of barcoding the samples because, of course, you stain everything together, you eliminate a lot of like technical variability, you run everything together. So also for batch differences is very, very useful, let's say. As I was telling you, you don't have to worry about spectral overlap, but still the methods that we use are not 100% pure, although the purity is over 95%. So what you have to do when you build your panel is to take into account the single spillover that you may have in close by channels. So what we do, basically, we test and titrate all our antibodies and making sure to choose a concentration where we can distinguish a negative from a positive, but it's lower enough to don't see any spillover in the nearby channels. And a little bit as for the flow, we have brighter, or let's say strongest in this case, channels than others. So the maximum ion detection is between 153 and 176. And this is something that you have to take in account when you're building your panel, because then these are the channels where you want to put your low abundant protein that you want to measure. So usually what we do, we tend to put in weaker channel surface markers that are highly expressed. And in a little bit in the strongest channel, we usually put phosphoproteins or even more transcription factors that are usually less expressed. The antibodies you can buy from the company that sells. The Cyto has also a patent proprietary, so they are the only one that can sell conjugated antibodies. They have a good portfolio antibody. They also have some panel kits, so a combination of markers to look at PB, peripheral blood, bone marrow, activation markers. So they have different type of pre-built panels. Otherwise, like often happen for us, you can conjugate your own antibodies for your marker of interest. So you buy a purified antibody from whatever company you want, and then you proceed with the method conjugation using basically a protocol that has already been established by the company. Just the antibody that you buy cannot be in any, so the carrier of the antibody cannot be a BSA or glycerol, because this will, let's say, have some problems with the chemistry when you do the conjugation. And then I added this, say something like Cyto cannot do miracles, because very often people ask me, oh, can I measure this or this or this? The truth is that if the antibody works, yes. If the antibody doesn't work or it's not specific, it's still an antibody-based technology. So still relies on the fact that the antibody that you're using is working, basically. The applications are very wide, as you can see here, and I just wanted to give you an idea of what I have used in the past, a little bit this technology for. Of course, immunophenotyping is the most used application. You can also look intracellular cytokines, intracellular phosphoprotein to look at different pathways, transcription factors. Even very interesting is also to add antibodies for specific cell state of the cell. So you can look at proliferation, cell cycle, activation marker. And then different groups, mainly at Stanford, I have to say, they have developed different panels based on their needs. So a group at Stanford developed an epitope panel to look at all the histone modification markers. And also they've been developing, also in my previous lab, some metabolic panels to look at different proteins that are expressed in different metabolic basic pathway. And this is something also that we are actually now implementing with the help of Angie, also here in our lab. As I was telling you, there is also all this. You can do it in cell suspension, which I have to say for us that we study a liquid tumor, it's perfect. But you can also do it in a slide. So if you're interested in studying solid tumor rather than the tumor organization, this is very powerful. because you can measure all these things on a slide and have an image with all this information, basically. But right now in the lab, we have only the cell suspension, let's say, type of instrument. We are missing the, this part, basically, that can be bought, and then the instrument can do both. Okay, so now I will give you a little bit, tell you how, in my experience, I've applied this technology to answer some biological questions, specifically related with the study of leukemia. Why we care about leukemia is because it's the most common cancer in children. And although you can see that in the last decades, the survival curves have achieved very, very good rates. So now we can cure way more kids than 50 years ago. Still, about 1 out of 5 children can experience a relapse of the disease. And unfortunately, when this happens, this disease is way more aggressive and way more difficult to treat. So the survival after the first relapse is still quite actually poor. One of the clinical challenges that we have right now is to try to really identify and intervene before patients experience relapse, before they have this basically more aggressive disease. Currently, and let's say leukemia, as many other cancers, it's characterized by a wide interpatient heterogeneity, but also intra-tumor heterogeneity. Thanks to the current clinical trial, we are very good in separating patients in different classes of risk. And this happens based on the clinical feature of the patients, biological features of the presence of specific mutations, and based on the detection of MRD, which stands for minimal residual disease. That is basically a surrogate of individual response of the patients to the treatment. However, this method, how we separate the patients, is still not perfect, because about 30–40% of the relapse happen in patients that we don't classify as high risk, meaning that we are still failing to identify some patients that actually can benefit of a more intensive treatment. And also the MRD detection is a pure quantitative analysis. It doesn't really tell us why these cells are resistant. And also doesn't really account for the intra-tumor heterogeneity, because all these analyses are performed in bulk, basically. The last decades, genomic studies have well shown how actually in the majority of the cases, the clone that is responsible for the relapse is already present at the time of diagnosis. And this is usually actually a minor clone, but can also be driven by the major clone that is present. So we actually, in our basic experience, said, okay, can we use a technology like this to identify from the very beginning which are the cells that are more associated with the risk of relapse? To do so, what we did was to analyze a cohort of 60 BLL patients, and this is exactly the work that as Angie was mentioning, I started during my PhD and completed during the postdoc. As you know, these are always long journeys, the publications. So we analyzed a cohort of 60 BLL patients with this panel, where you can see we had a surface marker on the left and a bunch of also actually a pretty good amount of intracellular marker. And of course, when we start to analyze this data, the first things that we realized that each patient was completely different from each other. When you put on a dimensional reduction plots, each patient starts to be in a different island. So it becomes very difficult to analyze. So we end up thinking about applying something that can help us to put all the patients on the same reference. So we started a little bit from what we know, that the patients have these BLL cells that are stuck in their development. So we decided to try to align all our samples on the B-cell trajectory of a normal bone marrow. And we basically developed this B-cell proteomic classifier. How it works is that we manually gate on a healthy bone marrow that we run along with our samples, all the population of the B-cell trajectory, starting from the hematopoietic stem cell down to the more mature B cells. Then for each of these populations, we extract the expression of 11 markers that we call phenotypic barcode, which are important to define all this population. And then we classify each leukemic cells to the closest healthy population. So by doing so, we basically were able to put all the patients on the same reference, let's say, and this was the results. So we had, of course, saw that the BLL patients had a higher expansion in specific population, and actually this was not groundbreaking science. It is how happened leukemia. So the cells are stuck in their development and they start to overgrow. So we could see actually where these blocking the differentiation was happening in our patients. And of course, the first thing that we said, okay, can we see differences in the abundance of this population between patients that relapse or not? And the result was actually no. So then we, again, one more time, decided to do things a little bit more complicated. So we decided to use all the information that we had, because as I told you, to build this classifier, we only use 11 of the 40 markers that we had. So we decided to extract all the features that we analyzed in all the cells and build what we called a deeper deeper predictor that stands for developmental dependent predictor of relapse. It's basically a machine learning approach. We divided our cohort into a group, a training and validation cohorts. What we did was, so the 80% of the training cohort, we did the tenfold cross-validation, and then we evaluate the model in the validation cohort, that is basic samples that were not in the training and never saw them in the model, basically. So by doing this machine learning, by using this machine learning approach, we identified 6 features that were associated with higher risk of relapse. And these features were actually, not surprisingly, all related with intracellular pathways that were activated in patients that were going to relapse in specific population. So this was an analysis that if we were performing bulk analysis, we could have not seen, basically. And this is how our predictor will separate our patients based on this machine learning, and where you can see that the red patients are the ones that relapse, and the blue right here are the ones that didn't relapse. So you can see that separates very well actually the 2 outcomes of the patients. In the same cohort, if we were using the gold standard or how we do it in clinic, were being separated in this way. So as you can see, we definitely adding intracellular information and running all these complicated analysis, we were able to better separate the real outcome of our patients. Of course, then we were curious about to see if the features that were present in diagnosis were actually persisting and relapse. So we analyzed a very small cohort of 7 patients, which we had diagnostic and relapse match samples. And basically we found the same feature that were still present there, and in some cases even more highly expressed. So after this, basically what we end up like, this is the graphical abstract. Basically what we found was that having at the time of diagnosis specific population with activation of basically were all downstream pathways of the previous signaling was correlated with a higher chance to have relapse. And as always in science, when you find something, that you have even more questions. So many more questions open up after this. First of all, we went to try to figure out how these features were selected, let's say, during treatment, if they were present in MRD cells, and even more, beside the activation-specific pathway, which were the transcription programs driving all this. And of course, if we could use all this information to better stratify our patients. So in the follow-up studies, we focus on glucocorticoid resistance. Why? Because basically glucocorticoids are heavily used during the first round of chemotherapy the patients receive during the induction therapy. Actually, they do a window of steroids and then they start chemo. And for years, we know that patients that don't respond to steroids are likely also to not respond to chemo. So we wanted to a little bit figuring out how this was happening. So we took 19 samples of the previous cohorts, and we saw the cells. We treat the cells in vitro with dexamethasone, which is one of the glucocorticoids used in clinic, dasatinib, which is another drug commonly used in clinic for a specific subgroup, and then their combination. And we run, again, a cytometry panel. This time, we tune it to have more information about the glucocorticoid pathway, how this was being regulated. And also, we wanted to figuring out a little bit more about proliferation of the cells and also how the cells were cycling, basically, and responding to the treatment. So the first step we did was taking the live cells after 48 hours from our site of analysis and run our classifier. And right away, what we noticed was that the cells that were surviving to the treatment were actually having a different phenotype. They were looking a little bit more mature. We wanted to dig a little bit more inside this, and we started using some cell lines that we treated with the drug. We ran RNA-Seq, and actually we saw that within the most activated pathway in cells that were exposed for 48 hours to the drug, the previous signaling was activated. And we also saw this by looking signaling protein that were downstream of the pathway. So as I said, we also use dasatinib, which is, even though it's a kind of dirty kinase because it's not super specific, where it acts is it's a dual SRC-ABL inhibitor. So basically, it's able to shut down all the signaling downstream of the PVCR. So by using these two drugs in combination, we could actually abrogate very well the signaling, both in cell lines and actually also here in our primary samples. But of course, we were interested to see if these two drugs together could kill better the cells. So we then performed some killing Nectin-5 staining with 7-AD by flow to test if the combination was synergic or additive. And what we found was that actually in the context of NAM6, which are more privy cells, the two drugs were acting in a synergistic way, while in the REH, which are cells that are a little bit stuck, a little bit early in the development, they are more propy cells, we had an additive effect. And when we combined the two drugs together, we were actually, even though it was just performing a couple of patients, to inhibit and kill the cells of patients that were completely unresponsive, treating the cells only for 2 days. So basically what we did, we took some unresponsive patients, we prolonged the treatment for up to 6 days, and then we could see major killing and also inhibition of the signaling. Finally, of course, we had to test this in vivo. We used an SG model where we injected NAM6 that were expressed in the luciferase, so we could monitor the disease using DLI. And here you can see how the combination of the two drugs resulted in a slower, let's say, engraftment and slightly increase in the survival of the mice. Okay, so then regarding resistance, in parallel, while I was in Cara Davis' lab, another colleague, Pablo, was working on another project that was actually related to a very important problem that nowadays we are facing. Because basically, patients that will go into relapse nowadays are eligible for CD19 CAR-T therapy, and in some cases also CD22. However, although, of course, we know that the best results of immunotherapy we have seen, we witnessed the best results of immunotherapy in the BLL setting, we know that not all the patients actually respond to this therapy. We still have about 50% of the patients that will end up also relapsing after CAR-T. And very often what happens is when leukemia come back, they lose the expression of the antigen that the CAR was directed against. So with Pablo, decided to use the same tools basically to try to figuring out if from the very beginning there were some feature that we could predict the patients that were going to relapse after CAR-T. I'm going to spoil you a little bit the results. We found actually a population of propy cells that even though they expressed the CD19 in the surface, they had lower level of Icaros, and this was associated with the negative, so with the relapse with the CD19 negative post-CAR-T. So how we did that, we analyzed, we were actually quite lucky to get a pretty big cohort of CAR-T treated patients, and this was done in collaboration with the Children's Hospital of Philadelphia, which were basically CAR-T were born, we say, in the group of Carjun. Then basically what we had was patient samples before and after CAR-T administration of patients that achieved complete remission, so the CAR-T worked very well, and patients that went into relapse. And we had both patients that experienced antigen loss, but also patients that relapsed with a CD19 positive disease, or even patients, as you can see here, they were completely refractory to the treatment. So Pablo, which is actually, I didn't introduce him, he's a great friend, of course, but also a colleague that he was working with me in Cara's lab. He's still there now, he's an instructor. He set up a panel of forensic marker to look at, to analyze all these samples. Again, we started from what we learned to do very well, which is aligning the samples using our B-cell classifier. And right away, we noticed that patients that were going to relapse as a CD19 negative post-CAR-T, they had already from the very beginning lower abundance of a pro-B population and higher abundance of what we call early non-B, which is a population that is not fully committed to be basically B. Pablo then decided to further analyze these pro-B cells in these patient samples. And what was actually very interesting is that the patients that were experiencing relapse had already, before the CAR-T, they expressed CD19, but they had very low level of Icaros. Same level as the patients after they see the CAR-T and they don't express anymore the CD19. And this was significantly different compared to the patients that instead achieved complete remission. So, of course, this was cool, but we wanted to dig a little bit more. So Pablo, which is a great biologist with also computational skills, he basically was able to run on a small cohort of these samples, a single cell RNA-seq analysis, performing using the BD-Relapsed platform, where you can basically analyze both protein and transcript at the same time. I skipped a little bit of part, but basically after a clustering analysis, we identified the probe cells. And what we found, we confirmed basically that these probe cells, even though there's still the CD19 in the surface, they have lower level of Icarus in the transcription factors. Then we look at which pathway were basically enriched in these cells, and as you can see, this patient had sort of enrichment of hematopoietic multipotent pathway, suggesting that basically these were cells that were a little bit more B, but not fully committed to BV. They were expressing also some transcription factor that were a little bit earlier in their developmental stage, basically. So then Pablo was able to actually nicely set up a bunch of tools to regulate and modulate the level of Icarus in vitro using BLL cell lines. What he did was create some knockdowns of Icarus. You can see very nice how when the Icarus goes down, it then knockdowns also the CD19 goes down. The first question we had, okay, is Icarus directly modulating CD19? And actually the question was no. There is another transcription factor which is well known to be correlated with certain expression, which is PAX5, but not Icarus. So we didn't find any difference between the Icarus wild type and Icarus knockdown cells in the chromatin accessibility of the CD19 promoter. But we performed when we did the GSA, one of the pathways that came up was RNA splicing. So Pablo wanted to a little bit investigate a little bit more this. And what he did was to try to apply a package to look at all the different possible splicing, RNA splicing that could happen, basically. And what he found was that patient and the Icarus knockdown cells, so the one that had a low level of Icarus, and also the patients that experienced CD19 relapse, had higher retention of the intron 10 of the gene of Icarus, basically. Sorry, of the gene of CD19. And in literature was already shown how the intron 2 retention is associated with a protein, CD19 protein, which is truncated and basically is not able to go to the surface and be expressed. Reason why basically the card don't see the molecule and they don't work. But the intron 10 part was never described before. So actually what we hypothesize here that this is a proxy event for the intron 2 retention that we actually also observed in some of our patients. And then, as I was telling you, since the patients receive also CD22 directly therapy, we actually wanted to look if also somehow Icarus was regulating CD22, and we could see also a decrease of CD22, although I would say it was not so impressive as for CD19. Then I skipped some of these parts, but just to say that then we decided to figuring out if cells that really had lower level of Icarus in vitro were less responsive to the treatment with directed immunotherapy. So basically, Pablo set up a Tigron system where when you add the drug, you decrease the expression of the transcription factor, and basically these cells lose the expression of the CD19 and CD22. And as you can see, when we give directed therapy, which is a monoclonal antibody directly against CD19, or CD19 CAR, which is a different type of costimulatory domain. We don't want to enter in the detail. Basically, the red dots that are the one that the level of Icarus tend to live to survive to the treatment. The same happened also when we treat the cells with CD22 CAR-T. So basically, this was published very I think a month ago, and also for Pablo was quite a long journey, but was nice and fun to work together in this project. Finally, I want to tell you how I arrived here somehow and what I'm doing now. As I was telling you, after all this work, we had some open questions, and one of the most fascinating for me was how to figuring out how these features that we see being associated with resistance are present in cells that are very resistant from the very beginning to the treatment. which is what we call MRD. And also trying to figuring out how this was related with the transcription programs of these cells. So as part of the proposal I submitted to IRC, what I proposed was to perform a multi-omic analysis of patient samples, performing cytometry, not only also transcriptomic profile and also clonality studies using single-cell DNA sequencing. And they like it, and they gave me the opportunity. They funded the project, and this is how I came back. Already told you a little bit what is MRD. This is just to say that basically what is performed nowadays is a quantitative analysis of MRD. What we are here trying to add to this is a qualitative analysis. So what these cells express, why they are resistant, and this of course can help in improving the risk stratification of the patients, but we hope also to identify some targets that can be used therapeutically for the patients. So to do so, we started from a cohort of BLL samples, 48 patient samples, which was a longitudinal cohort. Basically, we had from the same patients different samples collected at the time of diagnosis and after 8 days, 15 days of treatment, and 11 of these patients went into relapse. For 6 of them, we also analyzed the samples of the relapse. This time we went, sorry. This time we went even more crazy with I built basically a double panel, so 2 panels to look at not only leukemia but also all the immune population that are present in the sample. And basically we had, in this way, we could measure 80 markers, of which 12 were common between the two panels. Let's say a total of about 70 markers in all the cells. And as I was telling you, I used the barcoding system that I explained you before, because this was otherwise impossible. We had more than 300 samples total, with all the conditions to run it in a different way. And what I want to stress is that we added also in this time a healthy bone marrow, healthy peripheral blood as a control in our run. So this is how a healthy bone marrow will match in a dimension reduction plot. Here I used emblesome, is just one of the many. And you can see how nicely all the developmental population occupy all the space of the plot. And when we overlay over these our leukemic samples, you can see the leukemia is basically playing this space. Is, if you see before, basically it's the space of pro-B cells. So this is just a more fancy way to see what I showed you before with the classic plots. But what I want to stress here, of course, at diagnosis you have a lot of disease. When you start to analyze the age, the disease starts to go down, and for disease I mean the dots that are CD10 positive and CD45 low in this case. At the time of P15, if the patient is responding to therapy, you basically have almost nothing left in the bone marrow. So this becomes very challenging to analyze the few resistant cells that are there. So for this reason, we had to run millions of cells, and until the last drop of samples that we had, we did 12 hours run. It was very long, but somehow we made it to have a pretty good amount of cells also from patients that were responding to the treatment. So here is the representation of how our classifier will work on these samples. And again, a little bit, I don't know if you remember the plot that I showed you in the glucocorticoids part. You see that when in the very beginning, consider that during the first week, these patients receive only steroid, you start to see a shift towards a more phenotype used by the steroids. This was very similar to what we saw before in an in vitro setting, let's say. And then, of course, we also, in this case, we knew the outcome of the patients. We tried to see if there were any differences. Actually, this time we could find from the very beginning a different abundance of the probe cells. But when we ran analysis to look all the features, we cannot really easily separate patients that relapse from patients that don't relapse just by checking all the features in all the cells. So with the help of Ricardo, that joined my group in November, and he's now a PhD student in the DMET program. So we are now basically complicating our life a little bit, but it's funny because we're trying to develop a machine learning approach that this time doesn't predict the patients, or eventually will, but predicts the cells. So before, in what I showed you before in the Nature Medicine paper, we did a prediction at patient level. we care about the patients, if they were relapsing or not. This time, what we are trying to do is to predict the cells, because we wanted to figure out within the bulk of all the leukemic cells that we have in the diagnosis, which are the few cells that might be the famous minor clone that might be responsible for the relapse. So Riccardo has been, playing with this for a bit now. He tested a bunch of different classifiers. The one that performed the best, of course, I'm presenting today, which is the random forest. So the first step, what we did was to get leukemia. So we gated the blast, and then I handled all the gate to Riccardo, and we assigned the label in this way. So basically, we said if a patient is standard risk clinically or MRD standard risk and did not relapse, we consider cells from this patient as treatment sensitive, because eventually it's a patient that went well. If instead the patient went into relapse, no matter from, if he was belonging to a standard, intermediate, or high risk, we define these cells as treatment resistant. We assign this label, and then, and then we train a model in which, again, we separated the patients in a training and validation cohort. And also this time we did the leave one out cross-validation, but in this case, our leave one out basically was our 11 patients that went into relapse. So basically what Riccardo was doing was to train the model multiple times, training the model on the 10 patients that relapse, and then testing on the one that was not present in the training cohort, and this for 11 times. And then we perform a prediction on some of the samples of the cohort that we didn't use, specifically were all the patients that were in the intermediate risk group, and some of the diagnostic samples that were not present basically in the training cohort. These, I'm sorry for the text that is a little bit off, but anyway, these are the results of the random forest. We were actually, to arrive to this, we had to do some tuning. The model performs pretty well in identifying the true positives, so the accuracy is actually quite high, quite good, I would say, although we still suffer from some false negative cases, basically. So of course, we are working on this and trying to get as best we can, but overall, you can see here from the AUC is quite good. So this is sort of a model performance sensitivity of our model. And you can see that the mean AUC is 9092, means that in the 92%, let's say, we work. And of course, this is again patient specific. We have some patients that the model is very good in guessing will relapse, and some other patients that is not performing so well. Of course, this can depend on many factors. One of them is also the fact that not for all the patients we have the same number of events that we are training the model on. We tried our best to match the two classes with the same even number of cells, but for some of them we just don't have enough. Overall, what the output of the model is an extra. So together with all the cells, together with all our marker, we will have an extra column of prediction, which is another basic channel that goes from 0 to 1, where 1, of course, will be the cell predicted relapse, 0 the one where the predicted relapse. So we set up a threshold to 0.5, and when we look at the proportion of cells that are present in the high predicted cells using this threshold, to see if there was a difference between the patients that were relapsing from the patients that did not, you can see that actually the patients that went to relapse have a significantly higher percentage of high predicted cells, and this is independently from if standard risk, intermediate risk, or high risk. Based on the clinical feature. Here is just another way to represent the same data. You can see that the majority, so you can treat this if you see the top right dots, the patients that are highly predicted to relapse, and the one here are the low predicted to relapse. You can see the majority of our actual relapse, which are the red dots, end up in the highly predicted one. And this is just to say that it was very nice to see that we were able to pick the risk, the relapse, the intermediate risk, the relapse, and also the high risk. We also had, of course, some errors. Perfect. For instance, this patient is a standard risk, the relapse. We know that sometimes our cases that are very complicated. In this case, it was a patient that relapsed after 10 years. It's something that can happen, but it's very, very unlikely, let's say. Also, we had some patients did not relapse. Actually, they went to bone marrow transplantation because they had all the, let's say, clinically feature to relapse. So of course, for our model, these were there will be error in our model, but we know that these are patients that basically clinician did everything to make them to relapse. So it's something that we have to take in account, let's say. But basically, and I'm finishing soon. Just to say that, of course, we are doing all this because we care about the biology. We want to figure out these cells, what they express and why they are highly predicted to relapse. So now we are basically, with Riccardo, we are performing some supervised analysis of the predicted cells to figuring out which feature associated with relapse. Here we run the first feature importance. We've got some hits. Here just to show how basically we will be able to see in a plot now colored by prediction, high predicted cells from low predicted cells. And what we are trying now to do is to perform some clustering analysis, see which marker differential expressed between the clusters that are highly predicted compared to the cluster that are low predicted. Okay.
Attendiamo, proviamo ad attendere la risposta.
Okay. This is just, I'm almost done. Just to say that then within the next steps, what we want to do is to integrate all these site of data of the MRD cells also with the transcriptomic data that we got. So for 11 of the patients of the 48, we also run the RIP-seq, which is the transcriptomic platform of BD. We use a panel of 15 protein. So basically in this case, what you can do is basically you stain your samples with antibodies that are conjugated with oligo, and then you sequence the oligo. In this way, you can have the phenotypic expression of the marker, but then also look the transcriptomics, basically. In our case, we use exactly the same surface marker that we were looking, the phenotypic barcode that we were looking by CyTOF, to be able to then use this as a bridge of the two technology. And we know this is going to be very challenging. What we want to do is to perform a multi-omic integration of the two technology. Just a almost last slide, just to say that the nice thing of using cytometry technology is that you can take your cells, as you would do with flow cytometer or mass cytometer. So we get our blast, and then you can go and all the human to look at the transcription programs. So right now we are in a very early step of all this analysis. So hopefully soon I will give you more updates. And this is just to say last, really this time, that this is part of course, a big project. So we are now working the aim1, with the aim3, whole another aim very much focused on the glucocorticoid resistance, but this time looking metabolic rewiring of the cells, because we got some from the previous paper. We got some hints that something metabolic is also happening that is very interesting. So, and this is actually part of the things that we are going to start to work now with Martina, that just joined the group for her thesis. Nothing. Finally, I would like to thank everyone. Of course, my fantastic mentor at Stanford, Cara Davis, where I had a great time and we had the possibility to do great things together. We are having also fun in the meantime. And then, of course, my current institution, Fondazione del Tamanti and Professor Biondi, that they were very happy for me that, that I decided to come back and applying with them as a host institution. And all the people that are now helping me to build all this, of course, Angie, also with Sarah, we are running many projects together, and basically that's it. If you have any questions, I'm happy to take it. Mi sentiranno da casa, ma non fa niente. No, vabbè, no, va bene.
Thank you for this very nice work. I have just two curiosities. One is technical. So, in your site of experiments, which is your minimal, let's say, sensitivity limit to detect?
As number of cells, you mean?
Yes.
So right now, I would say you don't get to the same flow cytometer. That's for sure. just because the event rate, the fluidics is completely different. So you run less cells at the time. And also I would say 30–40% of the cells are lost in the process. So we start the process with 1 million cells. And then we...
I don't know whether you calculated whether if you have a very rare cell population, have, I don't know, 0.1%.
No, I can tell you that definitely you can arrive to 0.01%.
0.1.
Yes, yes. Our MRD cells were something like that at the day 15, for instance. So no, definitely, of course, what you have to do is to just run more cells. So let's say that the machine is able to detect this just in that case, if you want to look for a very rare population, you have to run instead of 1 million per patient, as we usually do, maybe you go up to 3–4 million to try to increase your sensitivity, basically.
And of course, that will be correlated to the expression of the marker they are going to use, whether a lot on the cells or not.
Well, that's, I'm not sure. Actually, no, even though, you mean the sensitivity is correlated with the, no, because actually, even if you have, I don't know, just 10 events, you can see very nicely the signals, basically, or even of low abundant protein, if this was your question, yes.
No, I suppose that you may detect a marker if it is very highly expressed on the cells, even though your cell is a rare cell.
Yes, of course. Yes, what I'm saying that even also the one that express lower level, you will still be able to see, because actually, if the marker is not expressed, you have a very almost zero background, it's less than 10. So you can nicely see even very few events that express even a low abundant protein.
Okay, and then the second curiosity I have is concerning the random forest, because
Okay.
I know it very, very well.
Ah, that's good too.
No, I mean, I was just curious, maybe you said and I just lost it. The settings that you used there were the observations, right? So you did the training
Cells, yes.
on the cells, and you didn't have for that experiment already the RNA-Seq data.
No, not yet.
Because
no, this is the next step. We want to definitely, once we figure out how to integrate the two technology and we are happy with that, definitely we can see if adding all that information will further improve the model.
So basically, the model trained on the 40 markers.
Yes, exactly. Yes.
Therefore,
yes, this is something that sometimes for all this modeling, it's a bit challenging because usually in the models you have more features than events. For us, it's the opposite.
Yes,
we have more events than features. So we know that it's sometimes a little bit challenging, basically, to find the right model for that.
It is also the number of samples that you are going to run with this model.
Yes.
Very well. You have a lot of samples and, as you said, more features than...
So yes, in our case, we have more cells than features, which is the opposite of what usually you get in RNA-Seq. Let's say when you run single-cell RNA-Seq, maybe you have 3,000 cells, but thousands of genes. So for us, it's the opposite, actually, yes.
So that you also were able to run the single-cell analysis on those samples that you selected.
We will, yes, it's in the to-do list, yes.
Yeah. Okay, so thank you.
And maybe we can chat more about it. If you are an expert, we are happy for any help.
Other questions?
Sorry, it was a little bit too long. I mean, mine was 45 minutes.
My question to you is about the cell, resistance. You can be already there, clonal, already there. And you can, there are very few cells, and you can mark on that cell.
So for this, yes. So what we do, so besides of how we identify these cells is based on the expression of marker that are actually used also to perform MRD by flow cytometry in as a gold standard. But something that also we want to run and was part of, I think, here of last this part of when I see clonality. So we want to run, so basically we have two things that we want to do. One is to perform with the transcriptomic data to go to look all basically the same rearrangements that you will look with the classic MRD that they do by PCR. And then also the clonality studies where this is a single cell DNA sequencing experiment where basically you can track the clone by mutation. So in that case, you can see if you have a clone that, I don't know, has a RAS mutation, then you have the follow-up time points, and you can see if that clone is still present or maybe has another mutation. So we actually ran one pilot experiment on just one patient where we had 4 time points, and it's actually very nice that you can follow the clone by the mutation they have, basically. And sometimes are meaningful mutations or driver mutation, but sometimes can also be not necessarily strong mutation, but it's still a good way to track the clones over time. The only problem nowadays of using this technology that is very, very expensive. So reason why we did the pilot, we'll see if it gets extra money to do it in a bigger cohort, basically.
Okay. Well, thank you very much.
Thank you.
For the very interesting seminar.
Ce l'abbiamo fatta.
Tu segui la mutazione?
Segui la mutazione o nel tempo? Praticamente
Non mi interessa tantissimo questa.
allora, però
Perché proprio la
è un po' è targeted, cioè tu scegli il gruppo di geni e anche già un po' tipo della leucemia, io non lo tipo 100